Bismillahirrahmanirrahim. Today lecture is about sense of smell. Why is smell so important? The sense of taste and smell allow to separate undesirable or even lethal food from those that are pleasant to eat and nutritious. The sense of smell also allow animal to recognize the proximity of other animal or even individual among animals. Smell also provide a warning for hazardous events such as fire or gas leakage from pipes. Sense of smell. Bears are thought to have the best sense of smell of any animal on earth. For example, the average dog's sense of smell is 100 times better than a human. A bloodhound's is 300 times better, and bear's sense of smell is 7 times better than a bloodhound's, or 2100 times better than a human. The olfactory system. The olfactory system consists of the olfactory membrane, olfactory receptor cell, olfactory nose, olfactory bulb, olfactory tract, olfactory striae, olfactory cortex. Output target of the olfactory cortex. The olfactory membrane. The olfactory membrane lies in the superior part of each nostril. Medially, the olfactory membrane folds downward along the surface of on the superior part of the septum. The olfactory membrane. Laterally, it folds over the superior turbinate and even over a small portion of the upper surface of medial turbinate. This is the superior turbinate, this is the medial, and this is the inferior turbinate. This membrane covers the superior turbinate and part of the middle turbinate. In each nostril, the olfactory membrane has a surface area of about 2.4 square centimeter. This area is about 2.4 square centimeter. Olfactory cells. Olfactory cells are the receptor cell of the smell sensation are bipolar neuron derived from the central nervous system. These are bipolar neuron. On one pole they have dendrite and on the other pole they have axons. They are bipolar neuron and originate from the central nervous system. There are about 100 millions of these cells in the olfactory epithelium. These bipolar neuron or olfactory cells are 100 million in number. The mucosal ending of the olfactory cell form a knob from which 4 to 25 olfactory hairs, also called olfactory cilia, measuring 0.3 micrometer in diameter and up to 200 micrometer in length, project into the mucus that coat the inner surface of the nasal cavity. These cilia, which originate from the knob, from each knob 4 to 25 olfactory hair cells originate, and they are projected into the mucus, which coat the inner surface of the nasal cavity. These cilia are covered by the mucus. Olfactory cells. These projecting olfactory cilia form a dense mat in the mucus. These cilia that react to odor in the air and stimulate the olfactory cells. Spaced among the olfactory cell in the olfactory membrane are many small Vomeronasal glands that secrete mucus onto the surface of olfactory membrane. These are the Vomeronasal glands, which are situated between the olfactory cell. This is the olfactory cell and this is the olfactory cell, and between these cells the Bowman glands are located, and it secretes mucus on the surface of olfactory membrane. And this is the knob, the ending of these dendrite and in a knob, and from this knob these cilia originate. Transmission of the olfactory signal into the olfactory bulb. How these signals from the membrane is transmitted to the olfactory bulb. The olfactory bulb lies over the cribriform plate. This is the cribriform plate, which is part of ethmoidal bone, and in the cranial cavity above this plate the olfactory bulb lies. The cribriform plate has multiple small perforations. This is the plate, and here are multiple small perforations through which an equal number of small nerve pass upward from the olfactory membrane in the nasal cavity to enter the olfactory bulb in the central cavity into the cranial cavity through cribriform plate. This is the olfactory membrane. From here neuron originate, and equal number of neuron in each perforation of the plate pass and end in the olfactory bulb. Glomeruli. Short axon from the olfactory cell terminate in the multiple glomerular structure within the olfactory bulb called glomeruli. These are short neurons which pass from the olfactory cell membrane through the cribriform plate and end in a structure. This is a structure which is called glomeruli, each of which is terminus for about 25,000 axons from the olfactory cells. In one glomeruli, about 25,000 axons from the olfactory cells ended here in the glomeruli. Mitral cell and tuft cells. Each glomeruli also is the terminus for dendrites from about 25 large mitral cell and about 60 small tuft cell, the cell body of which lie in the olfactory bulb superior to the glomeruli. These are the mitral cell located superior to the glomeruli, and from here the glomeruli also receive axons or terminals, and these mitral cells are about 25 in number. These glomeruli also receive small tuft cells which are 60 in number, and the dendrites from the mitral cell are 25 in number. The mitral and tuft cells send axons through the olfactory tract to transmit olfactory signal to higher level in the central nervous system. From this glomeruli and from the tuft cell, axons are sent to the olfactory tract, and from olfactory tract these signals are then sent to, or fiber are then sent to the higher centers. The trigone. The olfactory nerve around the area of the optic chiasma divided into three stria. This is the olfactory bulb. Here the olfactory nerve divided into three divisions, and this area is called the trigone. These are the anterior, medial and lateral stria. The olfactory nerve at the level of trigone divide into anterior, medial and lateral striae. The lateral striae travel to the ipsilateral olfactory cortex in the uncus. The lateral stria travel to the same side of the brain and end in the olfactory cortex in the uncus. The anterior stria cross to communicate with the contralateral olfactory cortex. This is the anterior stria and cross to the contralateral olfactory cortex. Additionally, it appears that the olfactory bulb talk to each other via the medial striae. The medial striae and the olfactory bulb talk to each other or share their information with each other through the medial striae, and a fair amount of neural processing seems to occur in the bulb themselves. Olfactory cortex. This cortex is not a single structure, rather it is defined as the combined area of cerebral cortex generally within the temporal lobe. This cortex is located in the temporal lobe that receive input directly from the olfactory bulb. It receives impulses directly from the olfactory bulb. This is the olfactory bulb, and when the olfactory nerve originates, it receives input directly from the olfactory bulb. This region includes piriform cortex, amygdala, and hippocampus. This is the hippocampus, and this area is the amygdala. Smell take a direct route to the limbic system, including the amygdala and the hippocampus, the region related to emotion and memory. The olfactory signal very quickly get to the limbic system. You can see this the olfactory bulb, and this is the olfactory cortex of temporal lobe, and the hippocampus receive a direct transmission from the olfactory bulb and also the amygdala. Stimulation of the olfactory cell. The olfactory cells are stimulated by the odorant, and on the surface of olfactory cell are present G protein. The G protein, when it is activated by the odorant, the alpha subunit is detached and it acts on the adenyl cyclase, which is activated, and the adenyl cyclase speed up the ATP and the conversion of ATP to cyclic AMP. The cyclic AMP then act on the calcium channels and open it. Due to this opening, the calcium enter into the cilia, and when calcium enters the cilia, it acts on the chloride channel, and chloride channel are open and chloride leave the inside of the cilia, and in this way depolarization of the cilia occur and the action potential is produced. The action potential travel down the axon of the olfactory receptor cell, eventually meets with the other axon and form the olfactory nerve, cranial nerve 1. Thank you. Please subscribe the channel and down the bell icon for further notification.